Welcome to Science Saturday, a new forum that highlights the important partnership between researchers and patients with complex lymphatic anomalies and their families. Science Saturday is sponsored by the Lymphangiomatosis and Gorham's Disease Alliance in collaboration with the Lymphatic Malformation Institute. My name is Mike Kelly, and I'm the Chief Medical Officer for LGDA and your host for today's session. The goal of Science Saturday is to highlight the impact of research on the care of our patients. The forum brings together patients, their families, and scientists, highlighting how research impacts the patient journey and how patient experiences help shape research. Today, we will discuss Brenna's journey, a young lady diagnosed with Kaposiform Lymphangiomatosis, or KLA. Brenna will share how her journey was impacted by exciting new research by Doctors Hakonarson and Dori at Children's Hospital of Philadelphia. The title of today's presentation is Kaposiform Lymphangiomatosis, Developing Personalized Therapies Through Research. Brenna, welcome to Science Saturday, and I want to thank you in advance for sharing your story. Please tell us more about you and how you were diagnosed with KLA.
Hi, thank you, Mike. So I basically had a healthy childhood up until I was about 9 years old, and I had this recurring strep that just wouldn't go away. Kept going back to the doctor, and it just was staying with me. And then on my 10th birthday, I got toxic shock syndrome. I went septic because of this bacterial infection that my body just couldn't fight. So now I'm in the hospital, and they find that I have these massive pleural effusions, these pockets of fluid in the interstitial space between my lung and my chest wall. And they were bearing down on my lungs. So I had 2 chest tubes placed on either side to drain this fluid. They stayed for about 2 weeks, and then they took them out and sent me home. And even though the hospital sent me home, I was never quite the same for like months after the incident. I was like, beforehand, I was able to just run and jump and play like any normal kid would. And then afterwards, it was hard for me to just like walk up my driveway without getting out of breath. So I definitely knew that something was still wrong. And I did physical therapy during this time. And the point where I went back to the doctor was I developed this bruise on my right breast. It was purple and blue, and there was petechiae, and it looked like I fell out of a tree. Really, like one of the doctors like just asked me if I fell out of a tree, and I was like, definitely did not come from the outside. And I went to a couple doctors before I got to the right doctor who saw it for what it was. They saw that it was a lymphatic anomaly pushing up from the inside. So at that point, I was admitted to the hospital, and I was there for months. Sorry, I lost my place. Hold on. So now I'm in the hospital for months on end, and as doctors looking at me, and they realize that I have a disease process, but they're not sure what it is or what to do. So now I'm going through these countless experimental treatments and biopsies, and I'm meeting so many different doctors and students, and they're all trying to take a look at me and figure out how to fix me. Some of the experiments that stood out to me, the treatments that I was on, one of them was they took me off food for 5 weeks and put me on TPN because they figured that somehow my digestive system was interconnected with my lymphatic system, and they thought the fat that I was consuming in my diet was contributing to the chylous fluid buildup around my lungs. But that didn't work because I hadn't eaten for 5 weeks, and I was still pumping out like a liter of fluid a day. So that didn't work. I was on interferon. I don't remember, does anyone know if that's an oral medication or if that like goes through because I know I had a Broviac line at one point, but I don't know what medicine I took.
It's an injection.
Okay, yeah. So I took interferon through a Broviac line. I remember at one point my lung collapsed. Thankfully, I was in the hospital when that happened. So they were able to inflate it right away. I remember one of the treatments they wanted to do was glue my lung to my chest wall and just get rid of the interstitial space. Thankfully, they didn't do that. I still have pleural regions. I was transferred to a bunch of hospitals throughout the East Coast. I went to Columbia Presbyterian, went to Boston Children's, I went to Cincinnati Children's Hospital, and then finally where I'm currently getting treatment is at CHOP at Children's Hospital Philadelphia. But the main medicine that I was on for a really long time was rapamycin or sirolimus. Am I saying that right? Yeah, okay. So I was on rapamycin or sirolimus, and that medicine helped me get to like a baseline okay, where it stabilized me, where I wasn't constantly accumulating pleural effusions. But for one, I still had sizable pleural effusions. Like I was walking around with a lot of fluid in my lungs and around my lungs. I had a chronic cough. I used a nebulizer as treatment, and I used the vest. A vest is like a commonly used treatment with cystic fibrosis. It helps with fluid in the lungs. But again, all these treatments were basically just taking care of my symptoms and keeping my effusions at bay, but I still periodically had to get fluid drained from my lungs. Like I was in and out of the hospital all throughout my childhood and all throughout high school too. And anytime I would catch a cold or bronchitis or pneumonia, which I would get a lot, my disease process would get progressively worse. And also with the rapamycin, I had to take supplemental meds like vincristine. So I was walking around with a PICC line for about 2 years, from 7th grade to about my freshman year of high school. And at this point, the toxicity of all these meds was really getting to me. And by the time I was in high school, I really lost my faith in like research and what they were doing on me. Like I really, like each consecutive year, I really truly didn't believe that I was going to make it to my next birthday. And I developed really severe anxiety and depression. And I was really inconsistent with my medicine at that point, because my mindset was basically, if this medicine is making me feel bad, I don't even know if it's working. Why would I take it? So then right after my high school graduation, I dipped again. I went back on rapamycin and vincristine and prednisone for about a year, and my hair fell out during this time, which was also very depressing. And I was at CHOP at this point. I was being treated by Children's Hospital of Philadelphia, and they started me on this new medication, trametinib, which is the most recent drug that I'm on. And I can't like rave about this drug enough. Like, it's honestly a wonder drug. I responded so quickly. As of today, I don't have a chronic cough anymore. I don't have pleural effusions. I don't have any respiratory symptoms. I'm able to run, like I ran a 5K in 2019, and I'm training for another one right now. And the toxicity of this drug compared to other drugs is honestly so minimal. Like the only side effects that I had was my scalp bled for the first 3 months that I was on it, but I don't have that symptom anymore. And the only thing that I'm really focusing on is making sure that I'm wearing sunscreen, because it makes my skin sensitive. But other than that, I'm basically able-bodied now. So I'm really grateful for trametinib.
That's a fantastic story, Brenna. Thank you for sharing. And I think that what you have experienced is very typical of what many patients with complex lymphatic anomalies experience. This convoluted journey, to make the correct diagnosis, and then an even more difficult journey to figure out what treatments will actually work, and how we can personalize those therapies in order to be successful in a particular patient. So I'm glad to hear you're doing great. You told me you were out hiking over the weekend, so that will motivate me to get out and do more. So I really appreciate you sharing your story.
Thank you so much. And I really appreciate this opportunity to share what I went through, because going through a chronic illness, any chronic illness or chronic pain, is a very isolating experience. That's why anxiety and depression are often comorbidities with chronic illnesses. And on top of that, having a rare disease like Kaposiform Lymphangiomatosis, when there's not a lot known about how the disease process works or how to treat it, you feel like no one in the world is really going through what you're going through. So I'm really grateful that I've been given this opportunity to open up and share and connect with other CCLA patients. And I appreciate that the LGDA even exists, because then it's like this sense of community so that people don't feel so alone in their experience. So thank you.
Thank you. Dr. Hakonarson, if you would please introduce yourself and to our audience and describe your role in the care of Brenna.
Yeah, thank you, Mike. So my name is Hakon Hakonarson, and I'm a physician scientist at Children's Hospital of Philadelphia and professor of pediatrics at UPenn. And I'm actually a pediatric pulmonologist by my medical training, and my PhD training was in molecular biology and genetics. So genetic research is really what I have conducted here at CHOP. I've been collectively at CHOP actually for about 25 years. Actually from Iceland, so it's a bit away, but we made a discovery a few years ago of some of the first mutations that underlie lymphatic disorders, and sort of gradually got to the mutation of identifying Brenna's mutation. And when we found Brenna's mutation, we could direct the therapy sort of through that precision knowledge. I don't know if you want to show the slides, like if you have those, if you go through. So I was just gonna sort of go through the discovery process, because this took many years. And the first family that we actually identified was a family. If you go to the next slide, this is just really to emphasize how we use genetic information to inform on therapy for lymphatic disorders. And the first family, it was actually a pediatrician at CHOP who had a son with a very significant lymphatic condition. And you can see that this was a 4th-generation pedigree where multiple family members were affected. So this was actually an autosomal dominant inheritance, and we identified the mutation being a splice-altering mutation in a gene called FRNB4, and FRNB4 is critically involved with lymphatic development. And you can see the proband, which is the sort of the black square here. The X-ray is very abnormal with a lot of fluid, and the lymphangiogram similarly shows fluid in the pericardium, in the pleural space, and in the belly. And you can see a very tortuous or abnormal lymphatic system in this patient. So if you go to the next slide, what we learned from this was to subsequently take that mutation into a model to study the effect of the mutation, to see if we could find any medication or drug that could sort of rescue or reverse this. And zebrafish is actually a very attractive model because it's a quick cycle. You can grow zebrafish in a matter of a couple of weeks, and you can study them. They have a lymphatic system, and interestingly, when we looked at this particular mutation, and because FRNB4 was a loss of function mutation, we could just knock out the gene in the zebrafish. And the zebrafish developed a very abnormal lymphatic central duct, misbranching, and sort of, pouches and swelling of the lymphatic system. And this is shown here sort of more schematically on the right graph. And if you go to the next slide, This slide actually shows the molecular changes that happen when you have a mutation in FMB4. And the most important one to look at is this second line on the plot to the left, where you see the P70 kinase. You see a P70 kinase being upregulated if you have this FMB4 mutation. This is where the signal is. And then if we use medications like sirolimus or like this BES235, both of it block the mTOR pathway, you can see that we shut down the P70 kinase and we can actually rescue or reverse this signaling upregulation in patients who have this particular mutation. So if you go to the next slide, but this was not the case for another patient who also had central conducting lymphatic anomaly like the first patient. So this patient actually had been on sirolimus, was a 12-year-old boy, and he had absolutely no response to sirolimus. And you can see the lymphangiogram here, similar tortuous sort of obstructive duct with misbranching, leaking fluid into the pleural spaces and elsewhere in the belly and the pericardium of this picture. But we identified actually for the first time in a lymphatic patient with CCLA mutation in this sort of RAS MAP kinase signaling pathway. So this was a gene called ARAF, and we found actually two patients with the exact same mutations and the same phenotype or same medical condition. And this mutation was highly, highly served across species. So we knew it was very important and most likely the cause. So if you go to the next slide, and when you see here is, if you look at the middle sort of plot, the P70 kinase is not changed at all in this particular mutation. But you see in the plot below and to the left that phosphorylated ERK, the p-ERK signal in the mutated state is highly, highly upregulated. And this is sort of interesting. You can see that very strong signal. And if you look at the plot here to the right up corner, you can see that these pathways, the mTOR pathway here to the left and the RAS-MAP kinase pathways or ERK signaling pathway that leads to ERK upregulation and MAP kinase upregulation is here to the right, and they are totally separate. So we wouldn't expect the medication hitting the mTOR pathway like sirolimus to have much effect. It can have some effect because there's always some crosstalk between them. So if you go to the next slide, we did the same sort of modeling work here in the cells, and we show that the expression of this gene called ARAF in the RAS-MAP kinase pathway was, if you had this mutation, you got this very sort of disorganized structure, which is the left lower corner of the plot with the wild type being on top. And then if you give a drug called trametinib, which is actually the drug that we ended up using for Brenna, you can see how that plot of the cell looks very similar to the wild type, to the normal state, because you reinstitute the expression of ARAF on the surface of the cell, and it's not like turning it on all the time to proliferate. And if you go to the next slide, here you see the zebrafish effects from using, a drug like trametinib. We used in this particular plot a drug called cobimetinib, just because the fish, they tolerate cobimetinib better than they tolerate the trametinib, but you have the exact same effect. And you can see how you rescue the abnormal branching, the central duct, which is very dilated and tortuous, normalizes in the plot on the bottom. You have the wild type on top, you have the mutation in the middle, and then you have the treated condition. And this sort of, suggested that this would be a drug that could actually treat mutations in the RAS-MAP kinase pathway. And if you go to the next slide, this is indeed what we did. Now, armed with the data from the zebrafish and from the cells, we actually got an approval from the FDA with a single IND use to take an adult medication, never given to a child, never given to a patient who didn't have cancer, and the IRB approved treating this patient here at Job, who was actually dying from his disease. And this was basically sort of, what led to that initial really, breakthrough of sort of discovery of the RAS-MAP kinase pathway in patients with, in a subset of patients with lymphatic conditions. And we published this in Nature Medicine a couple of years ago. And if you go to the next slide, here you see that there are not just the ARAP mutation, there are multiple mutations in the pathway that we subsequently found in other patients who upregulate that pathway. And we've now done the same job of translating these mutations into sort of innovative therapies for them. And if you go to the next slide, you can actually see that the mutation we subsequently found in Brenna, who at the time was about 18 or 19-year-old and obviously with a very significant disease, all of the tissues that had been tested before, the blood, saliva, skin, lymph node, they were all negative, because the nature of these mutations is that they are only present in the affected tissue. And we got a biopsy from a lesional tissue that led to the discovery of a 4% of the cells in that tissue being abnormal with a mutation. And unlike the ARAP mutation, this was a loss of function mutation with truncated protein that sort of lost function. So then you were thinking, well, that's going to be more difficult to treat. But then we realized we loses a negative control that upregulates the pathway. So the treatment would be the same. So that's how we came to trying trametinib in Brenna with her mutation. And you can see the validation of that mutation. Only a very few mutation spots were identified, but high enough to cause this medical problem. And Dr. Dori is now going to show the lymphangiogram of Brenna and her clinical course.
All right, great talk, Hakon. Let me just share my screen. All right, can you see this? Here, let me move this out. Good. All right, so again, thank you, everybody. Thank you, Mike, for arranging for this, and thank you, Brenna, for coming and sharing your story. I think this is extraordinarily important for people, giving people hope that that things can turn out this well. As I was introduced, my name is Yoav Dori. I am currently an interventional lymphologist, but started my pathway as an interventional cardiologist. And I currently am the director of the Gilliamore Fishman Center for Lymphatic Disorders. And my background is I started as an engineer, actually, and then became a physician, which has played a huge role in allowing us to develop some of the other tools, the other side of the tools of what Hakon was talking about, that have allowed us to diagnose and even treat some patients like Brenna, keep them stable until we can find the genetic mutation. And I'll introduce today a little bit about those things and the diagnostic tools that we have, some of the interventional tools, and I'll show you how we use those even in Brenna to assess her therapy. Just a second. There we go. So this is the system that we're going to be talking about, and I won't go too much into what lymphatics are in the lymphatic circulation, although a lot of you are probably familiar with this, but this is my schematic of what it is. And it is a circulatory system, was known as the forgotten circulation because we didn't really have a good way to image it. But in general, flow in the lymphatic channels are from the peripheral organs towards the center. And in most people, there's about 2 liters a day that goes through the main channel that is right over here in the center called the thoracic duct. that drains to the left side, the innominate vein or subclavian vein junction with the IJ, or the internal jugular vein on the left side. In some people, the drainage is to the right side. There's a lot of variability to this anatomy. But there are two very important streams that contribute to central lymphatic flow, and those are the liver lymphatics, which are shown over here, liver connecting to this sac that starts the thoracic duct called the cisterna chyli, and then the mesentery, and that is all the fluid that comes from when you eat fat. Mesenteric lymphatic increases, but this is the lymphatic channels that drain the gut. So these three streams of liver, mesentery, and the stuff coming from the lower part of the body and the other organs, most of the fluid from those streams ultimately end up in the thoracic duct. In addition to that, there is the lung streams that ultimately connect. As I said, this circulatory system was ignored for a little while because we didn't really have a good way to see it. And if, you know, and physicians said, if we can't see it, then we'll you know, then it's kind of not there. But luckily, that has now changed. And one of the initial imaging techniques that we use is what we call just T2 MRI. And that's shown over here. And this is an MRI technique that has been available for a long time, way before I started doing this stuff. That just looks at standing fluid, so fluid that's not flowing quickly. So you won't see blood, but you will see fluid, like you see over here, bright fluid in the stomach, and a little bit of fluid in other places. But this is what a normal image looks like, and you'll see in a little bit what abnormal looks like. But you can see that the lungs are dark. There shouldn't be any fluid in the lungs. And Brenna was talking about pleural effusions, and those will show up as fluid in the lung, and you'll see that in a second. But really, if we're dealing with a circulatory system, we have to have a way, and that's where my background in cardiology has really helped. Because as cardiologists, we talk about a circulatory system, we talk about the physiology of it, we talk about the anatomy of it, and we also talk about the flow in it. And in order to understand this lymphatic circulatory system, we really needed a method to allow us to image flow. And this is the imaging technique that we developed a while ago. It's called intranodal dynamic contrast lymphangiography, DCMRL. And this is what a normal intranodal lymphangiogram looks like. These are the lymphangiograms that Brenna has had, a couple of them. It's a very simple technique to do. We've now taught many people around the world how to do this. And the way it works is you just access lymph nodes in the groin and you inject MRI contrast agent and then image the system dynamically. And you can see the contrast here on the image on my right side, where it's going into the lumbar lymphatics and then ultimately makes its way up into the thoracic duct. In this case, there's a normal thoracic duct coursing towards the left and connecting to the innominate vein junction over there. This was a great technique, but this only shows us the lymphatic system that is attached to the lymph nodes and the central conducting lymphatic systems, but doesn't show us anything about the really most important, one of the most important streams, which is the liver, and the other is the mesentery. And so since then, we've developed the other techniques, which really allow us now to characterize everything, characterize the entire conduction system. And this is what we call intrahepatic dynamic contrast lymphangiography. So we access lymphatic channels in the liver and image what that looks like. And this would be a normal. So we're injecting in a needle in the liver. And you can see the retroperitoneal lymphatics are lighting up, and then the thoracic duct lights up, just like shown over here. This is this connection. And the last technique that we developed and just published about is this intramesenteric technique. And now this allows us to see the gut lymphatics. And that's extremely important for certain disease processes. And this is what that looks like. So mesentery, again, connects to this sac called cisterna chyli. This is the mesentery lighting up, connecting to the cisterna chyli, which is over here, which ultimately connects to the thoracic duct and goes to the left. So we now have the tools to basically image almost the entire lymphatic system. We can image the lungs, we can image the mesentery, we can image the liver, and that's extraordinarily important. And why is that important? Because it helps us to kind of phenotype the patients, and I'll show you what that looks like. So a lot of the patients with conduction problems, or KLA, which is the problem that Brenna had, One of the things that really hurts them is lung problems, is abnormal conduction into the lungs. And there's different ways by which that can present. It can present with leaking into the pleural surface, which Brenna had, which is fluid accumulation around the lung. It's called chylothorax if the fluid is chylous. But you can also leak around the heart, and you can also leak into the airway, causing plastic bronchitis. The imaging is really key in allowing us to determine what is the phenotype, what is exactly going on, how is this occurring, and then to be able to plan interventions. And I'll show you, for example, this is a child who has KLA, who we do imaging in, and this is the T2 imaging, and very different than the T2 imaging that we showed before, right? This lung here, you can see, is not dark, it's very bright. There is this is lymphatic fluid, actually. It's edema and lymphatic fluid, and you can see it's surrounding the airway. You can actually see the rings of the airway light up, which is not normal. And the lung is just very bright, and this is very abnormal, and this causes lung problems. And if you look at the dynamic imaging, you can see that instead of just a straight line going up, you have this flooding of the lung, which we call pulmonary lymphatic perfusion abnormality. This is a very bad conduction abnormality with a very dilated, tortuous thoracic duct. And when you have liters and liters of fluid just flooding the lung like this, you're going to have a lot of clinical problems. And this is what causes the chylothorax and everything else. But this is one patient with KLA. This is another patient with KLA. And you can see that the imaging in this patient is very different. And you can see that there is no thoracic duct, and fluid is crossing everywhere. It is conducting across the diaphragm, through the liver, actually going backwards in the liver, going backwards in the mesentery. And this is very different than the patient shown over here who has mediastinal brightness. There is fluid accumulating in the chest over here. But when we image the thoracic duct, you can see that the thoracic duct looks normal. There's not this perfusion of the lung from the thoracic duct. Knowing this is extremely important because this allows us to plan, first of all, how to try to help patients until we can get the genetic diagnosis using the interventional tools that we have now. And there's many of them. And again, this is all kinds of stuff that are under development. So one thing we can do for patients who have an abnormal duct, we can go in there and just shut it down. And this is what's called a thoracic duct embolization. This is putting a coil in the duct and then just gluing it shut. This is no longer really the preferred method that we use to do this because there's consequences to this, especially in patients with conduction abnormalities, because they will reroute. There's multiple other abnormal channels coming from all over the place. And if you stop the main channel for lymph flow, and sometimes you have to do that, this can have deleterious consequences in some cases. In some cases, it works really well. But there are other ways and new techniques that we've developed now to do procedures, and we can do selective embolizations. This is a patient with a bad conduction problem leading to abnormal perfusion in the lung. And you can see we have a catheter that's directly in the channel that was causing the problem. And you can see after the procedure over here, this is the thoracic duct. We leave it completely intact, shut down the problem, and this patient symptoms resolve. Now, these are the lung problems, and I said, as I told you, right, there's the liver and mesenteric components, and those play a huge role in some of the problems that we are seeing. And this is why the multi-compartment imaging is extremely important. And we now recommend multi-compartment imaging for everybody who has a conduction problem. This is the only way to characterize all the organs, the important organs, and see what's going on. But two other problems that patients present with conduction problems or KLA or any of the primary lymphatic disorders are protein-losing enteropathy and ascites. And to show you how the multi-compartment imaging is important, here is a patient who presented with a CCLA, bad conduction problem with bad PLE. And you can see we initially imaged the patient with intranodal imaging, and you can see that the intranodal imaging is completely normal. It did not show us what was the source of the problem. And when we did intrahepatic imaging in this patient, you can see that the imaging looks completely different. In this case, the liver is not connected to the thoracic duct, which you can see is coursing towards the left, but it is connected across the diaphragm to a duct that is actually coursing to the right. That is the right-sided duct system, which is supposedly a normal anatomical variant. But the fact that the liver is not connected to the regular central lymph system, that is very abnormal. But still, we did not see a leak that could explain why the kid had so severe PLE. And when we did the mesenteric imaging, you can see that really gave us the answer for what the problem was. And you can see that the mesentery, very abnormal, but not connected to anything. The mesentery somehow is isolated from the central conducting system, and all the mesenteric contrast was just going directly into the duodenum and just leaking in there. And this was causing the protein losing enteropathy. It was a congested mesentery, no way to decompress. And this allowed us ultimately to develop a therapy. So again, multi-compartment imaging is important. And like Hakon was talking about, now we have these phenotyping and we have the diagnosis. So we have the tools now to do diagnosis and actually characterize what the lymph system looks like, and have some tools to do interventions, selective or complete interventions to stabilize children. But in addition to that, it's extraordinarily important. Everything that Hakon was talking about, probably more important than anything else, is having the genotype, and a combination of these two things ultimately really allow us to do the therapies and stabilize patients and getting patients to be like Brenna, as having these targeted treatments. So in Brenna's case, right, when she presented to us, at that point, we did not do a embolization in her yet, but we were getting very close to do that. But she presented to us at the point that she was getting to be very, very sick and was very disabled at the time. Luckily, Hakon, thanks to Hakon's work, we were, he was able to find the gene, and we were able to start her on the drug, and the drug really has profound effects on the system, and I'll show you what that looks like in a second. So the first thing that we saw in her, and like she said, this was very quick. It's within a month, she had almost complete normalization of her pulmonary functions, going from about 50% to about 90%. And she's somewhere around there and a little bit improved. Like she said, she's running a 5K mile. She had a hard time walking for a short distance or even walking upstairs before. Her coagulopathy also got better. And she had complete remodeling of her central lymph system, which is what we see in these patients and is what is quite remarkable. And we didn't know that this was actually possible. People thought that if you had lung disease due to these abnormalities and you had it for a long period of time, then your lungs, that's it. You're going to have damage to the lungs, but that's not the case. And the lymph system remodels and quite remarkably and quite completely, actually. And this is her chest CT before procedure, and you can see there's fluid in there, and there's these ground glass opacification and thickening of the septi. So this is very abnormal, and this is what it looks like after therapy, and it looks basically almost completely normal. And this corresponds perfectly with our lymphangiogram. So this is Brenna's lymphangiogram before, and you can see the abnormal conduction in multiple places. She has a very dilated the tortuous duct, like the first patient that I saw with KLA before. And then there's just flooding of the lung, and this is liters and liters of fluid flooding of the lung. And post-therapy, the abnormal dilated duct is still there, but that's fine. The lungs have completely cleared up. She does not have any more flooding of the lung. There's complete remodeling of all the lung lymphatics, and that is translated also to her T2 imaging. So before intervention, you can see there's fluid in her chest. That's the pleural effusions that were causing some of her problems. But the lung itself, the lung tissue is very edematous, and there's edema everywhere else. And then after therapy, everything looks pretty much normal, and that corresponds with her clinical status. So using these type of imaging tools allows us also to assess how the drugs are doing and tailor the therapy. So I'll end with this and just saying that with kids with conduction problems or any primary really lymphatic disorders, this multi-compartment imaging is really essential in order for us to characterize everything, in order to guide the interventions and try to figure out, what to do, even to guide how to get biopsy and how to get tissue, understanding where the abnormalities are. That is extremely important. Selective and targeted procedures are definitely preferable, I think, than shutting down ducts. And really, it's the precision. It's the combination of both stabilizing patients, getting them better, and just giving Hakon's lab time to get us the genetics and get us the medical therapy that ultimately can create a Brenna. That's really the key in this whole story. And so I'll end there and go back to Hakon, and he'll summarize the rest of the stuff. Thank you.
Yeah, thank you. And just sort of, a couple of slides on sort of where we are heading. The key issue here is that the mutations, like the mutations that Brenna has, are acquired, and they are only acquired by a very small number of cells. And so it's very difficult to find them and uncover them. So we need to apply, the most sort of innovative approaches and methods to get to them. Because once we make a discovery of a mutation, we know how to treat. And that treatment could be treating through the mTOR pathway, treating through the PIK3CA kinase pathway, which is another sort of, pathway we didn't necessarily talk much about, or as with Brenna, where we see about 20 to 25% of the patients with more severe lymphatic conditions seem to fall into, which is the RAS-MAP kinase pathway, which previously was only associated with cancers. And now we see that a subset of patients with life-threatening disease, they have mutations in that pathway, and if we can find them and characterize them, we can treat them effectively. And if you go to the next slide, this just sort of shows where we are with that, because we have been treating individual patients like Brenna currently, but now we want to do really a full sort of fledged clinical trial. So we can get, because none of these medications are approved for children. They are all adult medicines that are only approved for cancer. So Dr. Adams, Denise Adams, recently relocated to CHOP, and she leads our clinical program and clinical trial development. And we are now going to move forward with a MEK inhibitor through a trial here at CHOP. And sort of, try to get the drug registered. And in parallel, because there are multiple pathways, we want to be able to offer patients from all over the world, no matter what pathway they have involved, we want to find it, and we want to design therapy that fits their mutation profile. And that sort of, is the personalized or precision-based therapy as we were talking about. And the ultimate goal is to sort of, have CHOP really being a hub for both national and international trials across this spectrum, and to guide therapies sort of, based on mutation detection. And we are scaling this now to multiple other sites, collaborative sites that we have, and seeking funding through the Zuckerberg Foundation and other foundations for this work. And just finally, on the last slide, we want to thank the families and Brenna. This would not happen if people would not be engaged and willing to do this type of research effort. So we are immensely appreciative of that, and funding resources through field atrophy, NIH, and LGDA and LMI. We've been fortunate to receive funding, in addition to some institutional support for the so-called frontier programs, which this is a part of. So with that, I'll close, and we'll be happy to take any questions if there are.
Thank you, Dr. Hakonarson and Dori, for sharing your fascinating research. Dr. Dori, I was not aware of the advances that you and your team have made in lymphatic imaging, or imaging the lymphatic system. This is critically important to genotype-phenotype comparisons to help us better understand the pathogenesis of the diseases that we treat. And as you're aware, we treat patients with CLAs, and these are overlapping diseases that we need to better understand the clinical and genetic features that really distinguish these diseases. So I think that your work will help us do that. And Dr. Hakonarson, this bedside to bench, back to bedside approach of treating patients is incredibly powerful. Where you're using preclinical models that you can manipulate in a way to better understand the underlying disease mechanisms and ultimately generate new therapeutics that can be used in the treatment of our patients. It's a very powerful very powerful research approach in order to personalize therapies for patients with all sorts of rare diseases, including complex lymphatic anomalies. So thank you for sharing that fascinating approach with us. And Brenna, thank you for sharing your journey. You know, you sharing your diagnostic odyssey and how you persevered is going to be very powerful for our patient population. And, you know, I'm glad you found the team at CHOP. And again, I'm glad you found a therapy that not only improved your quality of life, but allowed you to get back to the life as you want to live it. This is very reassuring to our patient population, and we really hope and we really thank you for sharing your journey and sharing your odyssey and sharing with us the terrific outcomes that you've seen so far. So thank you very much to all of you for participating in today's Science Saturday, and to our patient community. I really hope you have many comments once you've had a chance to review this fascinating segment. Have a great day.